Hello, and welcome to the Cancer Research Institute Cancer Immunotherapy and You patient webinar series. Today is Wednesday, June 24th, and the title of today's webinar is Understanding Genomics and Genetic Testing in Cancer Immunotherapy. Before we begin, I'd like to thank our generous sponsors who have made this webinar series possible. Bristol-Myers Squibb, with additional support from Foundation Medicine and Alkermes. My name is Arthur Brodsky, and I'm the Senior Science Writer at the Cancer Research Institute, a nonprofit organization established 67 years ago with a mission to save more lives by funding research that aims to harness the immune system's power to conquer all cancers. Now, it is my pleasure to introduce today's expert speakers. First, Dr. Corrie Painter is the Associate Director of Operations and Scientific Outreach, as well as the Associate Director of Count Me In at the Broad Institute of MIT and Harvard. She also leads the angiosarcoma project. Next, Dr. Eliezer Van Allen is Associate Professor of Medicine at Harvard Medical School, as well as the medical oncologist at the Dana-Farber Cancer Institute. He is also an associate member at the Broad Institute. So thank you both so much for joining us today. First, I guess, let's start by defining what we're talking about. So what is the difference between genomic testing and genetic testing?
Hi, everybody. At first, I also want to thank my friends in the cancer community who really helped us refine the questions that we're going to be talking about today, as well as come up with some of the answers that we'll be presenting later in this webinar. I really appreciate each and every one of you. We couldn't do the work that we're doing without your deep partnership and commitment to helping us further our understanding of genomics and cancer research. So, when I will say when I first started in this field, I'm both a cancer survivor and a scientist, I would ask this question all the time: what's the difference between genetics and genomics? And when I would first ask that several years ago, there was all kinds of mixed responses that I would get. Everything from they're the same thing, they're just nuances, to, oh, one clearly means, one thing and one clearly means the other. And it was pretty confusing. But over the last couple of years, I feel like the scientific community has kind of landed in a place where we all have adopted this similar way of referencing what genomics is and what genetics is. And you can think of genetics as all of the genes that are in your genome that you inherited from your parents and that you will contribute to any offspring. You can think of, the way that I like to think of this is that I have the same DNA in my cheek cells as I have in my white blood cells. And if you want an analogy, it's like they have the same book, and all of the words are identical in my cheek cell and in any other cell of my body. But the words that are read are different. Even though the words are the same, if you read different words, you're going to have a different meaning. And so you have different things happening in different cells, even though the underlying DNA sequences are identical. So that is what we refer to as genetics. Another word that we often use to describe this is germline. So the germline is what is in the egg or the sperm that gets passed on and happens to make you. When you have the same DNA in a cancer cell, things get a little messy. Cancer doesn't have all of the same safeguards that your normal cells have in order to make sure that you faithfully copy all the words in that book. And so things get out of control. The cells divide rapidly, they grow really fast, and they don't have the same proofreading mechanisms. And so you accumulate mutations over time, and those mutations might make the cells grow yet even faster and become more aggressive. So the DNA in the tumor is different than the DNA that is in your normal, healthy, or germline cells. And the DNA in your tumor is what we typically refer to as, like, when we test that, we're looking at genomic testing. So genetic testing is your normal cells that you inherited from your parents, and genomic testing is from the DNA in your tumor. So I'll leave it there. And if anybody has questions, you can pop them into the chat box.
Great. So, there's a lot of things that people might associate with genome sequencing or genetic testing. What is the role that these currently play in our health in general and in cancer specifically?
And so I can jump in on this one, and I'll say first, I actually want to also echo what Corrie said in the beginning. Thank you for giving us a chance to join this community and present today, or just have this discussion today. And also furthermore acknowledge, just, in light of everything that's been happening in the world, how much even more extraordinary the stress is for cancer patients and family and friends of cancer patients. And so for all those reasons, we're thinking of you, and we're happy to be here to be helpful. I also want to emphasize, I think what Corrie just outlined, an extraordinary way to think about genetics and genomics, and I know that much of medicine is coming up with jargon to confuse people, and so I'll try my best to keep to some simple terms, but I'll just so that in case you've heard these terms before, inherited, germline, genetics, DNA you're born with. Sometimes the genomics is referred to as somatic or tumor specific, or the other jargon that we tend to throw around, and I apologize on behalf of the field for that. In terms of how it's being used, in terms of our health in general, is as increasingly sophisticated ways. The inherited genetics, so again, the genes that you're born with, are now being used to find people who might be at higher risk for developing certain diseases. Some of those diseases include cancer, but not just cancer, and do so in all sorts of sophisticated ways. There's a lot of other use cases that are outside of health that genetics are being used for, lots of interest in ancestry and whatnot. In cancer specifically, it's actually been extraordinary to see how much genetics, again, the genes that you're born with, and genomics, again, the genes that are changed, the stuff that's changed in your tumor, have been used in cancer across the board. This is for everything from how do we pick what treatment or treatments to give patients or what not to give patients, all the way through to how do we find which cancer patients are not only at higher risk for certain complications, but also in whom we need to then talk about screening and their family members and everything in between. And then that's sort of that's been touched on in, not every cancer across the sun, but certainly just an extraordinarily large number of them, especially if we think about where the field was, even just 10 years ago. So I'd say in all sorts of ways, it's been quite impactful.
That's great to hear. And I want to stick with cancer, but first I want to just kind of dive in on one of those points a little deeper. So the genetic testing, which is the germline DNA that we're born with, and in our healthy cells mostly stays the same throughout our life. But what about the tumor DNA? We know that changes a little bit. So as that changes over time, does that mean that patients might require additional testing over time?
So that's a great question, and I think what you're describing is, or the question you're sort of posing is really an open one in our scientific field, which is, do tumors evolve over time? Do they, as they become resistant to the drugs we give them, does their genomic code change? And do we need to actually check multiple times to see how things have changed? The short answer is I don't think we don't know. We don't know in totality, and I think it'd be very hard to generalize. However, what we're starting to see across multiple domains is that it is indeed true that you can find genomic events in the tumor after certain treatments that were not present previously or not detected previously. And those events might be important for the next treatment decision. And so we can't say with certainty yet, but it's certainly plausible that the need to do genomic testing of the tumor more than one time will become part of a cancer patient's experience.
Gotcha. And so, in the context of cancer, perhaps the most well-known or popular use of genetic testing would be, as you alluded to earlier, testing to see if people have genes that might predispose them to cancer. And so maybe the most well-known one would be the BRCA gene, which can predispose people to breast cancer or ovarian cancer. But before we talk about, defining how genetic testing can be used to define someone's risk of cancer, I'd like to focus on how it's being used in people who currently have cancer. And you kind of alluded to it earlier, that after some of our treatments, the tumors can evolve, which can enable them to develop resistance against the treatments. So how is genetic testing being used or genomic testing being used today in the clinic to help improve care for cancer patients? And can it be useful for all types of cancer?
Great question. there's no one size fits all to that one either. But what I would say is that there are now enough drugs that depend on genomic events that can be found in any cancer, that it is, important to at least think about genomic testing of tumors really for all patients with advanced cancers. So, for example, there's a genomic event called microsatellite instability that has an approval linked to an immunotherapy that can be found, albeit rarely, in almost any cancer you can imagine. Looking for that is of interest. There's a few other examples of approvals of drugs that are tied to a genomic event that are not specific to a given cancer. And then on top of that, there are many examples of genomic events in tumors that are tied to specific drugs in specific cancers. So EGFR mutations in lung cancer, BRAF mutations in melanoma. EGFR and BRAF are just the names of two different genes linked to two different drugs, and it could go on and on and on. But there's a large and ever growing list of such events that is a lot of the reason what that ended up prompting actually Medicare and CMS approval of tumor genomic testing for patients with advanced or metastatic cancer.
So, sticking with genomic testing, what about with? I know that especially with immunotherapy, sometimes the tumors have been able to lose genes or gain, or mostly lose genes that enable them to escape the immune system's ability to detect it, and as a result, can make current immunotherapies like checkpoint immunotherapies less effective. But what about with respect to future treatments? Especially, as some cell-based therapies and some vaccine-based therapies are being developed that can allow doctors to target these unique mutations that are found in an individual's cancer. So where do how does the testing play into that today, or today and in the future as these are developed? So
it's a great question. I think for today, a lot of that work is happening in clinical trials. And in fact, there are even examples of what's called personalized cancer vaccine trials that are ongoing at multiple places in multiple ways. But what they're actually using is the tumor genomic information and what parts of the code, what parts of the book, that, as to borrow Corrie's analogy, have changed since the tumor became a tumor, and how can you actually develop vaccines specific to those events. So that's one example. Those are in studies. There's other examples with cell therapies that require knowledge about the pre-existing tumor genomics as well, and those have been used in certain cancer types already, and increasingly are being studied again for patients with a lot of other kinds of cancer, solid tumors and whatnot. A lot of that work is still sort of mostly in the clinical trial and research domain, although increasingly that's becoming part of what we hope will be sort of in our armamentarium, so to speak, going forward, really across cancer types. I don't know if Corrie, if you have any thoughts on that you want to add on as well.
I don't know that I have anything really to add on, though I did want to bring attention to some of the questions that are in the chat box that are kind of specific to this. We have one, Kelly was asking, is there a number for this? But I'm not entirely sure. Kelly, you want to clarify what you're asking there? And then another one is, and this is important and relevant to this point, is can the genetic testing help in immune therapy?
So it's a great question. And I think this comes back to actually something I know it's really worth emphasizing, which is, there was, as of maybe a year or two ago, an approval about a specific genomic event in tumors that can be found, that is commonly seen in some cancers like colon cancer, but can be found in almost any cancer. Again, it's called MSI, or microsatellite instability. Again, going back to the jargon is certainly a phrase that we have multiple different words for, but MSI would be the thing you could ask your physician about. And that is specifically linked to selective response to immune checkpoint blockade, to some of the new immunotherapies like pembrolizumab that are on the market. There was actually another announcement, I think it was last week, maybe even, about an additional genomic event, basically a count of a certain number of mutations that, again, are actually linked to some of the immunology behind how these immunotherapies work, that may further expand patients really across the board, regardless of one's sort of what type of tumor you have, and having that be something that could, be directly linking the tumor genomics to cancer immunotherapy. So that's a great question, and one I would say the field is moving so fast in that, there's late breaking news as of like a week ago about how fast this is moving. We think there's going to be more tumor genomic findings in time that will help understand these properties even better. But again, it's sort of, that's really at the cutting edge of this.
Great, and I think that's such an important point. You bring up the microsatellite instability and the levels of mutations that a tumor has. And with that MSI high approval, that is approved. Patients can now get the immunotherapy, I think, after they've failed the first treatment. But it doesn't matter what type of cancer they have, to your point, whether it's melanoma, breast cancer, brain cancer, if they have this tumor that's become genomically unstable, the evidence points that they're much more likely to respond to the immunotherapy, and they can now receive it. But also, Corrie, I know you've done a lot of work with the angiosarcoma project about how to kind of improve this. So what is currently I was hoping you could talk a little bit about what's currently being done research-wise to improve these genetic and genomic tests.
Yeah, I'm happy to. And, to everybody who's putting questions in the chat box, thank you so much. We'll definitely see what we could do to field everybody's questions. Keep them coming. So I certainly can only speak to the very small slice of the pie that I'm directly involved with. The research world and in general has a lot of different initiatives that are ongoing in order to improve those tests. What I'll say in general is that we don't understand a lot about what's going on in the DNA of cancers, in the cancer genome, just quite yet. And one of the ways that you could think about this is, we go back to that book. And if you just read all of the pages in a cancer's book, there would be all kinds of words that were messed up. And you wouldn't necessarily even be able to, you wouldn't know how to pronounce them. You wouldn't know what does it mean if you say it that way. And you would need to look at each word individually. But you don't have that ability when you look at in all of the genes, because they're all contributing to what's going on in that cancer at any given time. So you look at that book and there's like tens or even thousands of mutations that are all there. And you might not necessarily know which mutation is actually making that cancer grow rapidly, or which one is responsible for it to not responding to a drug anymore. In order to ask really detailed, specific questions like that, you have to take the research to another level. You have to be able to come up with model systems. Model systems are ways that you can look at the cancer outside of like a full body and try to understand each of the different aspects of the cancer that are contributing to the different things that make it grow aggressively or quickly. If we look at the different types of research that are ongoing, you can have everything from cancer cells that are like taken out of people and grown in a petri dish, and you might do stuff like put a drug on it and see what the effect is, and then look at how that might change the DNA, like Ellie had mentioned, evolve in the presence of a drug. Or you might do something like the research that Ellie and I are involved with through the Count Me In program, where we're partnering with patients in order to ask them to help us with the research. We ask patients to help us build these projects. The projects are aimed at understanding what's going on in the genes of cancer patients so that we can ask those specific questions. Once we build a project with patients, in my case, I built them with angiosarcoma patients, and in Ellie's case, he built them with metastatic prostate cancer patients. We can try to drill down on some fundamental questions by working with the patients in order to get their healthy germline or genetic-based DNA and their cancer or genomic DNA from their tumors, look at them and compare them to each other, and then also take into consideration, how's that patient doing? Did they have really aggressive cancer? Did they respond well to different therapies? And by looking at all of this data together, we can ask questions like, what are all of the mutations that we see in all of the patients that have angiosarcoma or metastatic prostate cancer? What is the impact that these mutations have on the rapid growth of the cancers? Do people with different mutations in their DNA respond differently to therapies? And does cancer DNA change when people are on therapies? Meaning, like, does the DNA evolve and therefore escape the effects of that therapy? If we can ask all of these questions, we can then feed those, the answers back into those tests, so that instead of getting reports that say variant of unknown significance, which is what a lot of people will get when they have their DNA report from what's a panel testing or genomic testing, they'll get that set of words back that says, basically, we in the scientific world don't quite understand what the significance of the mutation is. And we're sorry about that, but you have it. And so all of this work we're doing, not just us, but the entire research world, is trying to converge on a world where we know what all of the mutations do, and also what therapies we can give if you have specific mutations.
And I actually want to follow up on that, because I think, what Corrie's partly getting at is, I think, some of the answers that I've given already, which are, to summarize, we don't know yet. And there's so much we don't know. And I think, part of the reason we don't know is because in our sort of traditional way of doing these kinds of studies, we require patients to come to us in a very few select places around the country or the world, and to ask these questions. And so, whether it's the cancer vaccines, the cellular therapies, some of the other discoveries, the idea behind Count Me In, whether it's the angiosarcoma project and the metastatic prostate cancer project, there's, an ever-growing list of them under the Count Me In umbrella, is that we can now actually bring the research to you, and you can actually join this movement wherever you are. And I think the hope is that we can cast a much wider net to really actually answer all of these questions more directly. I'm following the public Q&A section here, and there's some extraordinary questions. I think a couple of them have been, what is the genomic state of the art for breast and prostate cancer? And those are two diseases for which there's a lot of movement, although the immunotherapy movement has been slower in those diseases than in others, outside of those two exceptions I mentioned. And that's actually a lot of the motivation for the metastatic breast cancer project and the metastatic prostate cancer project, which is simply, we just don't have enough data to even know. And so it sort of further reinforces that this space we're in now, this sort of genomics and genetic testing and cancer immunotherapy, is so far in front of the cutting edge of where the field is, that the community here, have an opportunity to actually drive it. Patients and family members, and the community itself is really actually going to be key to pushing this forward.
Yeah, and I think that's a great point. What you both have mentioned about how there's still just so much we don't know. I think back to, when the human genome was first published in 2003, and a lot of people thought that was kind of the answer to a lot of things. And it was really just kind of the step zero almost, that, we're learning so much more about how not only all of these genes interact, but the genomes which genes are turned on in which different cell types and how all those interact. A number of other factors that I could go on and on about, but I will stick to the genomics for now. So, traditionally, the only way to really look at a tumor's genome was to take a piece of the actual tumor out. But now we're getting a little bit past that. And there's some terms that are starting to become a little more relevant that, still might not be fully understood. So I was hoping we can go into some of those. So first, what is a liquid biopsy? And can you define the terms CF and ctDNA?
Yeah, sure. So liquid biopsies are a relatively new phenomenon, which holds so much promise for us to understand what's going on in people's tumor DNA without actually having to do a biopsy of their tumor. So the word liquid biopsy really is just a fancy word for a blood draw. And so the hope and what we're seeing is that tumors can release DNA from inside the tumor cells into your blood. Another word for that is called shedding the DNA. And so if you take a blood draw, there's a chance that you may be able to actually see the tumor DNA floating around in the blood. Because it's in circulation with your blood, sometimes it's referred to as CT, for circulating tumor DNA. So that's ctDNA. There's also regular normal cells that also release DNA into the blood as well. So if you draw blood from any person at any given time, you're going to be able to detect some DNA in there, whether it's from healthy cells or from malignant cells. So if you look at just all of this, the DNA that's in circulation, another way that scientists refer to that is CF, for cell-free DNA. So a liquid biopsy is really just a blood, it's also referred to as a blood biopsy, but it's really just a tube of blood that's taken, and the DNA that's in that blood that's not encapsulated by any cells, that's just floating freely, is able to be sequenced just like a tumor, just like the tumor DNA that would be extracted from somebody's tumor. And in that way, you have a non-invasive way to go and try and take a look at what's going on in somebody's tumor just by taking a vial of blood.
And what would be some of the advantages of that? Of being able to look at it through the blood?
Yeah, there's several advantages. First, just not having to go and get an actual biopsy. So it's much safer, much more comfortable. A lot of times, something that we underestimated was that for patients who are undergoing constant therapy, like in the metastatic stage where they have ports put on because they have ports put in because they have treatment at regular intervals, it's very easy for that port to be accessed by the person who's setting them up for their therapy anyway. And so for them to contribute a vial of blood to have a testing like this done is much easier than even in some instances providing saliva. And so it's an easy way for people to be able to get that DNA, and you can get snapshots of what's going on in tumors over time, because you can look at, you can draw blood at a much faster, greater intervals than you can actually getting tissue directly from a tumor. So you can look at what's going on in that evolution that we were talking about by taking serial blood biopsies and looking what's happening over time. Though I don't think that that's like a commercial use of it just quite yet, but I could see it moving in that direction. And then lastly is that you, instead of getting information from one tumor that you may biopsy, there could be several tumors, if you are in the metastatic setting, that all shed and contribute to your understanding of what's going on across multiple different tumors.
Gotcha. So before we move on real quick, I see that maybe we should spend a little more time discussing the microsatellite instability and tumor mutational burden that we brought up earlier. Would you guys be able to maybe clarify that a little bit more for the audience?
Sure. Yeah. So it's confusing, and just in case you're wondering, it's confusing to everyone, and to many of us, many of the providers. So you're not alone. So microsatellite instability is a phrase that refers to a certain kind of change in the tumor genetics that results in a very specific pattern of changes across the entire genome, and that pattern is very distinct. And so it has its own name, microsatellite instability. It's oftentimes associated with just having a lot of mutations, a lot of changes in the code. And though having so many of those changes, again, has this complex relationship with the immune system. And for many reasons is why immune therapies tend to work really well in patients with microsatellite instability, which again is this sort of genome-wide change that has a very specific pattern and results in a lot of mutations. That's one very specific category of a broader space of genomic changes that can be under a category of high tumor mutational burden. So that simply refers to mutations of any cause. It could be from microsatellite instability or a host of other reasons. And there's a thought that a similar biology is associated with sort of more immune responsiveness in patients with high tumor mutational burden, or TMB. That's sort of the acronym that's thrown around a lot. And those patients have a similar enhanced response to immunotherapy. They're similar, but not the same things. You can have patients with microsatellite instability who, for a host of reasons, actually have a low TMB. And you can have patients with a high TMB who do not have microsatellite instability. the first FDA approval for immunotherapy was actually linked, that was across cancers, was for microsatellite instability. The one from last week was for high TMB. If you're confused by that, just know you are not alone. We all are, because we're all trying to wrap our heads around this and figure out how to make sense of this in real time, because again, it's moving so quickly. I don't know if that was helpful, if I made things worse, but I'm happy to try to take another lap around that one.
I guess I think one of the important things that I'd like to point out, is, obviously some of these mutations drive the cancer. They give them the ability to grow or resist the immune system or just survive and thrive in general. But another really important point, especially in the context of immunotherapy, is these mutations, these genetic mutations ultimately lead, or in some cases lead to proteins that are now abnormal and look foreign to the immune system. And so that is one of the main reasons that the more mutations someone has, the more likely there's to be these mutated proteins that then the immune system can recognize as foreign and respond to as if, the immune system, when it sees foreign, it doesn't know if it's from a virus or a bacteria or a cancer cell. So the more of these mutations it has, the more potential targets for the immune system to go after it.
Yeah, maybe I would like to just take one little minute and expand on that also, because it's really kind of fundamental to understanding genomics and immunotherapy in general. So, I'll stick with the same analogy. You have a book and it has the words in it, and something has to read those words and turn them into something that has meaning. The meaning of the words are the proteins in your cells. And if they're wrong, if they're read wrong and they look wrong, just like Arthur said, that your immune system has been trained since you were born to crawl through your body and basically look at what is you and what is not you. And everything in your immune system is geared to protecting you. And so it will totally ignore all of the genes that turn into proteins that it recognizes as you. But the second it sees something that's not you, that it was never trained to recognize, it is going to try and kill that thing. If it's inside of the cell, it's going to kill the cell. If it's outside of the cell, it's going to shoot antibodies at it and basically just try to get rid of it. So when you have a cancer that has either microsatellite instability, or in the case of angiosarcoma or melanoma, where the sun is beating down on the DNA and causing mutations, in either case, if the end result is that the DNA in the tumor has a lot of mutations, that's going to lead to proteins that your body was never trained to recognize. And just like we heard, if it looks like it's not you or self, your immune system can go and attack and kill that cell and or try to take care of that tumor. So a lot of what we're talking about and why these drugs have been kind of FDA approved is that in these tumors that have a high proportion of proteins that are different than you, the chances that you can use a checkpoint inhibitor, which is just a drug that will take the brakes off of the immune system and allow the immune cells to go in and eradicate those cells that carry these different looking proteins, is higher across all the different types of cancers. And it's a super unique way that we can combine the genomics plus the one drug across a variety of different cancers.
I actually, and on that point, there's a question here about if you have a low tumor mutational burden and you are not microsatellite stable, does it mean immunotherapy will not work? And I just want to very emphatically state the answer to that is no, because these are not perfect, what's called biomarkers, or which is a word that defines the way we try to figure out which patients should get what drugs. There are a lot of other considerations to be made. There are entire classes of cancers. I'm thinking of kidney cancer, which is one of the ones that I treat, that has no such thing as microsatellite instability or high TMB or tumor mutational burden, but has a lot of patients who respond to immunotherapy in a similar pattern to lung cancer, bladder cancer, melanoma, and so on. So I just want to be very emphatic and state that just because you have a tumor genomic test that does not show these things, that does not mean you are not a candidate for immunotherapy. That's a discussion you got to have with your physician, because there are many nuances to that and a lot of moving parts that are happening in real time.
Absolutely. So now sticking with, I guess, the practical considerations for patients today, how is this testing actually done and who is eligible? And does insurance cover the cost?
So the testing, if it's a test off of the tumor biopsy itself, that's a test that your doctor can order, and that's facilitated through your doctor and the hospital system. Those tests, there is a Medicare approval for this. I mean, obviously, every insurance company is going to have a different coverage pattern. If it's a liquid biopsy test, again, it's a similar kind of procedure because it's a simple blood test. It can be done sort of through your hospital system. I would say that the insurance coverage is probably going to be a little bit more complicated there, and I think it's a conversation you should have with your medical team as to sort of the specifics about you and whether those things are covered. But then those things go off to the companies, and the test comes back to your doctor, ideally, typically within a few weeks.
And as we've been talking, a lot of this still is very cutting edge, especially the liquid biopsies. We're still trying to optimize them and determine where they can be most valuable. What about patients on clinical trials? I know that the cost of drugs are covered for there. Is it possible that the cost of the biopsies might be covered under there for patients on clinical trials as well?
It should be. I suppose I can't speak for all clinical trials. I would say that we certainly do our best to think about all of those considerations up front and make sure that those costs, any costs that might be sort of attributed to the study, are covered by the study. It's not a perfect system. I'll be the first to admit that, but sort of every effort is made to try to make those coverages possible.
I can appreciate definitely a complicated system.
Yeah.
So now I'd like to go back to at the beginning. I mentioned, I think in the popular consciousness, the most well-known form of genetic testing is to see if someone has the BRCA gene or something else that might predispose them to cancer. So could you talk a little bit more about how this is used to determine the risk of cancer in healthy people? And also, how do consumer testing kits such as 23andMe, where do they come into play here?
It's a great question. So again, just to reorient everyone, we were just spending most of our time talking about tumor genomics or somatic genomic testing. I think we're pivoting back to inherited genetics or the germline genetics. To go back to Corrie's analogy, that's the book, your book of life. About
the original copy.
Not the changes in the tumor. The way it's being used now is it's I'd say in a few different capacities. It's in patients who have a very strong family history of cancers, who may not have cancers themselves. They might be referred for, again, germline or inherited genetic testing to look for a cause. If a cause is found, that might lead to different screening. So they might be, if they find something, they have more MRIs done frequently or other kinds of tests, colonoscopies, and so on and so forth, to try to find cancers earlier than they would for other folks. And similarly, for patients who have cancer and who have these inherited events, it's relevant for two reasons. One, it's relevant for so-called cascade testing. So that's a phrase that means, if they find it in you, then they at least want to present the conversation about whether any family members that you're related to you specifically also want to be tested. Sort of that's the cascade of testing, so to speak. And then likewise, there's increasing evidence that some of these inherited genetic changes, so not the changes in the tumor, but the changes that you were born with that actually impact the tumor, have treatment impact. And some of the same concepts we were just talking about in the tumor also apply in the germline or inherited genetics. So microsatellite instability and immunotherapy, BRCA1 and 2, and certain other drugs. There's a lot to consider there, and a lot of ways it's being used. To your question about some of the consumer testing kits, I'd say some are comprehensive and some are not. I would strongly advise patients to talk with their medical team to try to navigate what tests or tests are appropriate. Because even for some of the vendors that offer these tests to the consumer specifically for cancer screening. There are actually different flavors that they will offer. So, for instance, they have like prostate cancer specific ones or colon cancer specific ones, and so it's a nuanced conversation that you should definitely have with your treating providers. And don't assume that a consumer test that you might have done for some other purpose is going to be comprehensive for cancer screening.
Gotcha. And I do want to just go follow up on one point you made with respect to the microsatellite instability, and that there are some people can inherit genes that may make them more predisposed to that. And one prominent example is Lynch syndrome, which could you explain a little bit about what that is and how it plays into it?
Oh, and also before you do, can you fold Leaf-Romani in there as well, since we did have a question specifically, and they may be able to be coupled together in the answer.
So, right. So again, inherited genetics are the changes in your DNA that you're born with. Some of them are very well known and have been sort of described and sort of named by certain people who made these discoveries. So have different names about them. Lynch syndrome, in a lot of respects, encompasses the genes or the changes that are the things that cause microsatellite instability in tumors. Those same events can happen in the tumor and are not Lynch syndrome. So I guess here I am going around in circles, but that is a very specific clinical type of a clinical situation that we think about that is distinct from other germline or inherited genetic syndromes like Leaf-Romani, which involves a different gene and has a different set of considerations associated with it, both for screening and for potential treatment. I don't know if that, maybe I'll stop there and see if that addresses. Because just to be clear, Leaf-Romani and the P53, the gene that's related to it, is not the same. Is not part of the same things as Lynch syndrome, which is not part of the same thing as other diseases. And again, it gets there's just because the genome is so vast and there's so many changes that can happen. Each of these things has a different pathway associated with it.
And from my understanding, a lot of these genes, they don't all do the same function, but from what I understand, they all kind of intersect at a cell's ability to repair its DNA. Is that correct?
For many of these things that are involved in inherited genetics that lead to cancer predisposition, the common sort of point of biological sort of relevance is the ability to repair your DNA. Yeah, it's just that I don't want to say that too narrowly, because I know that whole thing is just a massive, a massively complicated system. So yeah.
Corrie, I didn't know if you had anything to add before we.
No, I just, I guess I would say that different syndromes may contribute to those same things we were talking about before. For example, Lynch syndrome often will lead to a high tumor mutational burden. And therefore, people with Lynch syndrome may, though they don't have other characteristics that, or have cancers that are like traditionally thought to respond to immunotherapies, may indeed have a good response. And so again, really talking with a genetic counselor as well as your physician, if you have any of these predispositions, to see if that's playing a role in the genomics of your cancer and whether or not that then would make you a good candidate for immunotherapy, I think is really important. And so a big take home is whenever you have questions, to really dig deep with your physician, and if at all possible with a genetic counselor.
So are there any drawbacks or safety issues associated with these tests, or maybe any reason someone wouldn't want to do them?
Yes, and I will. So I'm going to take that question, and I want to give a little bit of a preamble though before I dig into this. I went ahead and turned to my cancer community, and when I say that, I mean really in social media, I'm connected with cancer patients across so many different types of cancer. I've been an advocate for over 10 years after my own diagnosis with angiosarcoma, and have worked closely with the metastatic breast cancer and prostate cancer and esophageal, stomach cancer, brain cancer, other sarcomas. And so in my kind of social spheres are patients that are kind of representing from all of these different cancers. And I ask them this because I feel like the answer to this is not just like a set in stone answer. A lot of this is perceived risk and what people have experienced themselves. And who better to answer this question than cancer patients? So I do want to say I have a variety of answers from patients, and I don't want to you just like put this out there to scare people, but I think it's absolutely important for everybody to learn from each other. And so I'm going to go ahead and kind of read what patients said when I asked them this very question. There was a variety of different answers. Several people said to me, I have no concerns at all. In fact, it brought me great peace of mind to be able to have a sense of what's going on in my genetics. And I had a great genetic counselor who went over all of the risks associated with any of the things that popped up in my results. And it was really good. And so that's like one end of the spectrum. No concerns at all. Really happy that they did it and felt like they had peace of mind as a result. And then there were other people who shared other things to consider. More than one patient had concerns that their employer would gain access to their data and discriminate against them. Luckily, we have laws in place to protect against this. But folks are, they're concerned that laws can change. Their DNA is not going to change. And if their DNA has been put out there and somebody owns that DNA and the laws change, what happens to them? And along those lines, could they be discriminated against with respect to insurance? Could their children be discriminated against in any of these domains in the future as a result of them getting tested? Because you do share your DNA with your relatives. So what is the impact if somebody owns your DNA code and they do something nefarious to yourself and to other people around you? I think that's mostly theoretical because we do have laws in place right now, but it's a concern and something I think people should certainly be aware of. Another concern is and this came up a lot actually, and it was written in different ways, but ultimately, people had concern about the anxiety, about knowing too much about their predispositions. And what do you do with that information, especially if there's nothing you can actually do, if there's no treatment and you just have this kind of hanging over your head that there's something in my DNA and I can't do anything about it, but it's there. And now I'm just sitting there waiting and waiting and waiting. And so some people felt like that was something to give them tremendous anxiety. And other people thought for the exact same reason that it was empowering to know. And so what some of the people that responded to me said, and they're so right, it's just such an individual choice to do this. It's an individual choice, but to go in and understand what you're getting involved with is so critically important. And the best person to help you think through that is a genetic counselor, because they will tell you before you get the testing done, these are the risks to you knowing this is what if you did have, something in your DNA, what we could do about it, and so on and so forth. So it can help you understand what you're getting involved with if you do have that level of anxiety. Another important point that I wanted to put out there was somebody brought up, and this kind of piggybacks off of what Ellie was saying, that there could be a false sense of safety if receiving results from a consumer testing company. A consumer testing company may not give, they may sequence one tiny little part of one gene and say whether or not you had a mutation there or not. But that doesn't necessarily mean if you don't have something that you're out of the woods. There's those things that we talked about, those variants of unknown significance and or other parts of the same gene, which may be equally as concerning, but that the consumer test just didn't even look at. And so if you get that test and it looks like you're good, you may not go and really monitor closely things that if you never had that test, you would be more prone to take a look at because you might feel like you're safe. And then the last one, which kind of, fleshes out a little bit what I said at the beginning, is somebody was concerned with who owns my data and what does that mean? What are the implications? Are they going to sell my data? Are they going to make money off of my data? And so really, breaking it down, privacy concerns, other people knowing what the results are and discriminating against you, ownership concerns, and the personal reflection of what you would do with the information concerns. And then I think I'll stop there.
I think it's an extraordinarily well sort of put together set of concerns that I think I will just say on personal anecdotal experience have seen come up in conversation over the last 5 to 7 years as it's really snowballed clinically in different ways with different patients. And again, would just emphasize that my hope should be is that you should not feel like you have to make these decisions and thought processes alone, and that there's trained professionals whose job it is exclusively to help you navigate these precise issues and make the right decision for you, which is going to be a very personal one.
Yeah, great points by both of you. So now, kind of on the background of what we've been talking about, that it's still kind of early in our understanding of all of this. So how do doctors make sense of these results, and how accurate are they?
So that's a big question. I would say, certainly 5 to 7 years ago, most medical oncologists were not that comfortable with these test results, and especially the tumor genomics, because again, the inherited genetics were being handled by genetic counseling teams, that were very experienced with this. But for a lot of us in the cancer community, we were sort of given these reports. They just started coming, and a lot of folks were perhaps not entirely sure what to do with them. The good news is that a lot has happened in the last 5 to 7 years. The content and the reports themselves have improved significantly to sort of from the companies and the vendors that provide these tests to help guide clinicians on how to interpret them correctly, and how to consider a lot of the sort of nuance that I think Corrie was mentioning that the patients have brought up about, how do we know it tested the right thing, or did it look at the right part of the tumor, or was there a technical problem, and so on and so forth. And so doctors have gotten a lot better at that, and similarly have gotten a lot better at understanding which of the sort of list of things that they see can actually be acted upon more directly, and how to best consider which things might be relevant for a clinical trial or might need to be explored in some other way. So I'd say there's been a lot of improvements. Although I'll be the first to admit, and even this is true in my own practice as somebody who's entire academic career is basically studying this, that I still sometimes struggle interpreting some of the results in individual patients because the results are either confusing or contradictory or unexpected for any given patient. And I think that just speaks to the sort of real-time uncertainty that we have in this field. We just don't know enough about enough people from enough different places, and with enough different experiences to confidently act on a lot of the stuff we get in these reports. In terms of the quality of the tests, I would just defer to our regulatory bodies have a few. There are some tests that have gone through the sort of like extremely rigorous testing evaluations to make sure that they're vetted, and those are the ones that tend to get reimbursed by insurance companies. So so long as one is sticking in that realm, I suppose those are the things that I would think about.
Gotcha. So we're almost out of time now, but I was hoping that each of you could leave us with your kind of final thoughts and what you think are the most important messages for patients.
Dr. Painter, I don't know, you want to go first? Or?
I've said it a couple times, but I do see a lot of questions popping up. There's still so much to know and learn. I'm sorry we could not get through and address all of the questions. Maybe we could do a follow-up at some point and really drill down and dig into a lot of the concerns and questions that people have. I think the take home from me, just based on that alone, looking at the chat box, is that we have a long way to go in terms of bridging the gap between what we know and what we don't know on the scientific side, and what patients know and what they want to know on the patient side, and that we are at a really unique time where we can come together and address these things. I would say in the interim, when you have these questions, really leverage your medical team. Don't hesitate to reach out and demand answers. Not that you have to demand anything, because I'm sure everybody's there to help you, but really don't hold back. I think people have a tendency to think, I only have 5 minutes with my doctor, and so I'm going to, just like hear a couple of questions. But my advice is to draft all of these questions that you have, and, just keep asking your medical team to meet with you until you have these answered. Meet with genetic counselors too, because they're just an excellent resource that I personally didn't utilize until I was like 9 years into my own personal cancer journey. And I had so much, I was on the side that had peace of mind after talking with them and learning about my own risk factors and the risk factors that I was likely to pass to my children. So that's my takeaway, is that we have ample time to come together and learn from each other more as patients, scientists, and doctors, and that we should all be leveraging our medical team for the most amount of information we can get.
So I couldn't have said it better myself, so I won't try. I will say, I'm approaching the 10-year anniversary of moving to Boston to start my medical oncology training. And in the summer of 2010, I remember this because I remember me and my co-fellows being laughed at by some of the senior physicians about two things. One, whether we could do genome sequencing at scale in cancer patients to help guide treatments, and two, whether we could actually harness the immune system to treat cancer. 10 years later, it's almost, the field is unrecognizable compared to where it was when I started not that long ago. And those two things we almost take for granted, that it's so it's almost taken for granted now that we have these incredible immunotherapies that we have that are so effective for so many patients, that we can do genome sequencing in all sorts of complicated ways and use that to guide patient care, and that we're really actually moving the field forward. And so there's profound optimism. And I would just say that the real way to take the next major leap forward is that patients are going to have to, really they're the ones that have the power. You have the power to actually make the field sort of take these incremental but good steps and turn them into major leaps. Because at the end of the day, we still just don't know enough about most people in most cancers, with most settings and most drugs. And so until we figure out how to do that as a community, I think, we won't be able to make that big leap. So my hope is that we can all continue to come together in venues like this to have these conversations and really take a big leap forward together.
Great. Yeah, we covered a lot there, and I hope that our audience found it as enlightening, as informative as I did. So for more of our webinars and the additional resources that we have for patients and caregivers as part of CRI's Answer to Cancer educational programs, we encourage you to check out our website at cancerresearch.org/patients. Here, you can read and watch stories by others who have received immunotherapy treatment across a wide variety of cancer types. You can register for one of our virtual immunotherapy patient summits, browse our entire library of past webinars featuring the world's leading immunotherapy experts, and access information on other resources, including treatment, emotional support, and financial assistance. You can also find help locating an immunotherapy trial that might be right for you. Finally, I'd like to thank our sponsors one last time for making our webinar series possible: Bristol-Myers Squibb, with additional support from Foundation Medicine and Alkermes. And I'd also like to recognize and thank BioRender, whose platform is used to create the title graphics today. So thank you all so much for your attention today. I hope you found today's webinar very informative. And again, you can watch this and all of our other webinars on our website at cancerresearch.org/webinars to learn more about immunotherapy options in a number of cancer types. Dr. Painter, Dr. Van Allen, I just want to thank you both so much again for taking the time to speak with us today and for the amazing work that you are doing to improve care for patients. And I'd also like to thank our audience, who was extremely engaged today, and I hope you all enjoyed it. Thank you.
Thanks so much.